Welcome to this lecture on computational biology. Today, we are going to explore one of the most fundamental and powerful concepts in biostatistics, the confidence interval. Whether you are estimating the height of a plant species or testing a life-saving drug, understanding how to measure and report uncertainty is absolutely essential. Let us dive in and see how we can make reliable scientific claims from limited data. To begin, imagine you are trying to guess the exact number of jelly beans in a massive glass jar. If you just give a single number, like exactly 452, you are almost certainly going to be wrong. However, if you say that you are fairly sure the number is somewhere between 400 and 500, you have a much better chance of being correct. This intuitive idea is exactly how a confidence interval works in statistics and biology. Instead of trying to pinpoint an exact single value for an entire population, we provide a safe range, or a net, that we believe catches the true value. Now that we have an intuitive grasp from our jelly bean analogy, let us define this formally. In biostatistics, a confidence interval is a specific range of values calculated from a sample of data. This range is highly likely to contain the true unknown population parameter. Remember, the true population mean is a fixed number out there in the real world. It is not a moving target. It is our calculated interval that changes with every new sample we take. Therefore, a confidence interval gives us two crucial pieces of information at once. It provides our best estimate of the true value, and it gives us a measure of how precise that estimate actually is. Building on that definition, we must understand what the percentage attached to these intervals actually means. When we say we have a 95% confidence interval, we are stating our level of trust in our sampling method. It means that if we were to repeat our exact experiment 100 times, taking a new sample and calculating the interval each time, about 95 of those calculated intervals would successfully contain the true population mean. The confidence level, which is usually set at 95% or 99% in biological research, tells us how reliable our methodology is over the long run. So why does this statistical concept matter so much in computational biology and medicine? In the real world, it is physically impossible and financially ruinous to measure every single individual in a population. You cannot test a new cancer drug on every human on Earth. Instead, scientists test the drug on a small sample of patients and calculate the average response. But they need to know if this sample average is a lucky fluke or a reliable reflection of how the whole world will react. The confidence interval bridges this gap by telling researchers the margin of error around their sample data, proving whether a treatment is genuinely effective. Before we look at the math, let us identify the three pieces of information we need to build this interval. First, we need our best guess, which is simply the average or mean of our sample. Second, we need to know how spread out our data is, which is represented by the standard error. Finally, we need a multiplier that represents how confident we want to be. This multiplier comes from the standard normal distribution. When we put these three components together, we create a powerful formula that adds and subtracts a specific margin of error from our sample mean. Now that we know the components, here is the mathematical formula that brings them together. The confidence interval equals the sample mean plus or minus the margin of error. Let us break down every symbol. The x-bar represents the sample mean. The z is the z-score, which increases if you want a higher confidence level, making your interval wider. The S represents the sample standard deviation, showing individual variation. The N is your sample size. The entire fraction, S divided by the square root of N, is the standard error. Notice that if you increase your sample size N, the standard error becomes smaller, which makes your confidence interval narrower and much more precise. Let us apply this formula to a real scenario. Suppose you are a botanist studying medicinal plants. You sample 100 plants and find an average height of 50 centimeters, with a standard deviation of 10 centimeters. You want a 95% confidence interval, so your z-score is 1.96. First, we calculate the standard error by dividing the standard deviation, 10, by the square root of the sample size, which is 10. This gives a standard error of 1 centimeter. Next, we multiply this by our z-score to get a margin of error of 1.96 centimeters. Finally, we add and subtract this from our mean of 50. We are 95% confident the true forest average is between 48.04 and 52.96 centimeters. Let us look at a slightly more complex medical example. A company tests a blood pressure drug on 64 patients. The average drop is 14 millimeters of mercury, with a standard deviation of 16. They need 99% confidence, so the z-score is 2.576. We divide the standard deviation, 16, by the square root of 64, which is 8, giving a standard error of 2. We multiply this by our z-score to find a margin of error of 5.152. Subtracting and adding this to our mean of 14 gives our final interval. Because this entire interval is well above 0, the company has strong statistical evidence that the drug genuinely lowers blood pressure. To truly cement this concept, let us look at it visually. Here you see a standard normal distribution bell curve. Right in the center is our sample mean, representing our best estimate. Extending outwards to the left and right are the margins of error, which establish our lower and upper bounds. The large shaded area in the middle represents our 95% confidence level. Notice the small tails on either side. These represent the remaining 5% of uncertainty, split equally into 2.5% on each side. This diagram perfectly illustrates how the confidence interval casts a symmetrical net around our sample data to capture the true population parameter. Having seen the visual representation, we must address a critical trade-off you will face in biostatistics. Notice what happened in our blood pressure example when we demanded 99% confidence instead of 95%. To get that higher confidence, we had to use a larger z-score. Mathematically, multiplying by a larger z-score forces our margin of error to become wider. This means our final interval is less precise. In statistics, you are always trading precision for a higher guarantee of capturing the true value. If you want a narrow, precise interval, but also want high confidence, your only option is to drastically increase your sample size. We have covered a lot of ground today, so let us summarize the key takeaways. A confidence interval is a powerful statistical tool that provides a realistic range for an unknown population parameter based on limited sample data. It openly acknowledges that no sample is perfect and mathematically quantifies the uncertainty inherent in biological research. Remember that the width of this interval is controlled by the interplay between your sample size, the standard deviation, and your chosen confidence level. Whenever you read a biological study, looking at the confidence interval will tell you not just what the researchers found, but exactly how much you can trust their findings.